In cryopreservation of large-scale tissues and organs, fast and even rewarming is necessary to minimize ice formation and prevent temperature gradients across the organ during rewarming. Both of these can damage cells, whereas the latter can cause physical cracking of organs when rewarming, as shown here. Nanowarming is a novel technique that addresses this issue by increasing the rewarming rate through usage of iron oxide nanoparticles, which are evenly perfused into organs to be cryopreserved. Iron oxide nanoparticles are commonly used as heat sources via irradiation by an alternating magnetic field in the radiofrequency range. The alignment of the magnetic moments of the iron oxide nanoparticles, as well as the physical movement of the particles as they rotate to align with an external field, produces losses in energy as heat. By perfusing iron oxide nanoparticles into organs to be cryopreserved, we can distribute the particles evenly within the vasculature alongside cryoprotective agents, vitrify the organ, and prior to transplant, irradiate the organ with radiofrequency to rewarm evenly. To ensure even, fast heating, particles must be evenly distributed within an organ and well dispersed in solution, meaning not aggregated to ensure heating rate isn't affected and blockages within vasculature do not occur during perfusion. Quantification of iron oxide nanoparticles and their distribution becomes important as concentration is directly proportional to the warming rate at a given frequency. This helps determine the ideal settings for rewarming to ensure cryopreserved organs are rewarmed quickly enough to prevent ice formation and organ cracking without damaging tissue. Because iron oxide nanoparticles are magnetic, common methods used to quantify their concentration are via nuclear magnetic resonance, or NMR, and magnetic resonance imaging, or MRI. Both of these modalities commonly detect how the magnetic moment of ensembles of protons in water are affected by their surroundings. A typical MRI or NMR experiment manipulates these spins using external magnetic fields to determine the environment of these protons based on how the magnetization of protons in water changes over time when perturbed by one of these external fields. The magnetization can be perturbed and measured using the following base experiment. A static magnetic field in the z-axis, called B0, is applied to align all proton spins along the z-axis. This field is typically very large, on the order of 1 Tesla or higher. A separate, much smaller field called B1, transverse to B0 in the xy plane, is applied at a specific frequency called the Larmor frequency, typically equal to the field strength times the gyromagnetic ratio of protons in water, to excite spins and tip them into the transverse plane, where a receiving coil can detect the spins. As the B1 is turned off, the spins relax back towards B0. The rate at which the spin Z component relaxes back to its original value is called T1 relaxation and is related to the local environment of the protons in water. T1 of a proton in water can be related to the concentration of nanoparticles, as the local magnetic field produced by the nanoparticles shortens the T1 relaxation rate in a concentration-dependent fashion. So more particles means a higher local magnetic field experienced by the protons in water and therefore a shorter T1. T1 can be measured by either inversion or saturation recovery pulse sequences, which generally estimate T1 by applying B1 along a user-specified sequence of timed intervals and recording the signal generated for each time to generate a curve of magnetization recovery. The fit for this curve is generally an exponential, with T1 as the rate constant. Practically, most NMR and MRI scanners will have a saturation recovery or inversion recovery sequence pre-built into them, and you'll just need to enter in the time ranges that would work for measuring the full range of relaxation times to obtain the T1 of your sample. It is important to set the recycle delay, aka the time spent waiting for the spins to relax back to B0, to be roughly 5 times your T1 to ensure the spin is finished relaxing. T1 is also field dependent, so different NMR or MRI instruments may need different T1 versus nanoparticle concentration curves. NMR is used to measure bulk T1 of a sample and therefore gives no spatial information and typically measures small samples, but has great sensitivity to small concentrations of nanoparticles. Typically the reciprocal of T1, aka R1, is used for establishing nanoparticle concentration mapping curves. To actually quantify the nanoparticle concentration from an R1, the iron concentration, which is proportional to the nanoparticle concentration, needs to be confirmed with another method, such as inductively coupled optical emission spectroscopy, or ICP-OES. ICP-OES can quantify the iron content in a sample directly, and is a gold standard for iron quantification. However, it is also destructive. Using the iron content gained from ICP-OES, a given R1 can be mapped to a concentration of iron, typically in milligrams iron per mil sample. This must be repeated for a set of varying concentrations to establish a linear range for nanoparticle detection. MRI has less sensitivity but can spatially resolve nanoparticle locations to an extent. As shown in this T1 weighted image across the murine hindlimb, the nanoparticles show up as black voids in the image. When the nanoparticles are evenly distributed locally, the T1 decreases, so the signal intensity increases, causing the pixels to appear lighter. However, the concentrated clusters of particles can produce local field gradients even within a single pixel. Because of this, pixels are displaced from their true locations, resulting in the signal void shown here due to signal pileup and signal dropout. Fast imaging sequences such as SWIFT or swept imaging with Fourier transform can be used to measure exceedingly short T1s produced by high concentrations of particles. However, high particle concentrations can also lead to blooming artifacts like these, which can influence spatial resolution. Hopefully this video is a good introduction to iron oxide nanoparticle quantification for cryopreservation. If you have any questions, please feel free to email me. Thanks.